you. When you have a really important molecule like DNA that is ultimately responsible for controlling the cell, it would make sense that when you make another cell, like in mitosis, you would also have to get more DNA into that cell. And that introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where. It occurs in the nucleus. If the cell has a nucleus, remember, not all cells have a nucleus. This video clip is actually going to focus on the types of cells that do have a nucleus. They're known as eukaryote cells. Prokaryotes, which are cells that do not have a nucleus, they do things a little differently. They also do DNA replication, but that is not going to be our focus for this clip. Next, when. When does this happen? Well, this typically happens during a stage known as interphase. Interphase is when a cell is growing, it's carrying out cell processes, and it's replicating its DNA. You know what it's not doing at the exact same time? Dividing. You don't want a cell to be replicating DNA and dividing at the same time. That's a little bit too much multitasking. So DNA replication does not happen during cell division, otherwise known as mitosis. In fact, cells replicate their DNA before division processes like mitosis and meiosis. Because once you make the new cell, you better have DNA to put in there. I think DNA replication would actually make a great video game. It's actually quite exciting. I'm going to introduce the key players in DNA replication so that you can get some background information. Now, the majority of these key players that I'm going to introduce are enzymes. In biology, when you see something end in ASE, you might want to check, as it's very possible that it may be an enzyme. Enzymes have the ability to speed up reactions and build up or break down the items that they act on. So here we go with our key players. First, helicase. This is the unzipping enzyme. If you recall that DNA has two strands, you can think of helicase as unzipping the two strands of DNA. Helicase doesn't have a hard time doing that. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, poor DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer that it makes? It's actually a piece of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you would need that a little later. Now don't feel overwhelmed. We'll go over this sequence in order. Please keep in mind that like all of our videos, we tend to give the big picture, but there is always more detail to every biological process we cover. That's really true for DNA replication. DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. There can be multiple origins within the DNA. At the origin, helicase, the unzipping enzyme, comes in and unzips the DNA. SSB proteins, that stands for single-stranded binding proteins, bind to the DNA strands and they keep them separated. Primase comes in and makes RNA primers on both strands. Now remember, that's really important because otherwise when DNA polymerase comes in, it wouldn't know where to start. Now DNA polymerase can get to work. Remember, it's the important enzyme that adds DNA bases. Now, you have two strands, right? They're not identical. Remember, they complement each other. They also are anti-parallel, so that means they don't really go in the same direction. With DNA, we don't say a strand goes north or south. The directions for the DNA strands are a little different. We say that DNA either goes 5' to 3', or 3' to 5'. This can seem very confusing. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of the ring structure. Now you do the same thing for the other side, but keep in mind this strand is flipped, just because DNA strands are anti-parallel to each other. So let's count these. Again, clockwise from the oxygen, 1', 2', 3', 4', 5', and the 5' is out of the ring. This strand on the left runs 5' to 3', and the strand on the right here runs 3' to 5'. Well, it turns out that DNA polymerase can only work in the 5' to 3' direction. So the strand that runs 5' to 3' is fine. It's called the leading strand. But the other strand will make it a little tricky. DNA polymerase can only go in the 5' to 3' direction. So RNA primase, it has to set a lot of extra primers down to do that, as shown here. It takes longer too. This strand is called the lagging strand, which is pretty fitting. On the lagging strand, you tend to get these little fragments of synthesized DNA. They're called Okazaki fragments. Amazing name. The primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, remember that's the gluing enzyme as I like to nickname it, has to take care of the gaps in the Okazaki fragments. Now at the end, you have two identical double helix DNA molecules from your one original double helix DNA molecule. We call this semi-conservative replication because the two copies each contain one old original strand and one newly made one. One last thing. Surely you have had to proofread your work before to catch errors. Well, you definitely don't want DNA polymerase to make an error. If it makes the wrong DNA base, then you could have an incorrectly coded gene, which could ultimately end up in an incorrect protein, or no protein. DNA polymerase is just awesome. It has proofreading ability, which means it rarely makes mistakes. And that's a good thing. Well, that's it for the Amoeba Sisters, and we remind you to stay curious. Sometimes it feels like DNA gets all the credit. Yes, DNA is very important. It codes for your traits. But sometimes what gets left out is how important RNA is. Because without RNA, you actually couldn't get that genetic message out to your cells so that they can start producing proteins. RNA is a very important biomolecule, just as important as DNA. So what we're going to do right now is compare and contrast RNA with DNA. This is really important to understand because if we don't understand it, we can't understand protein synthesis. And protein synthesis is the process that gets your DNA to code for the proteins that make up so many of your traits. So let's talk about DNA and RNA. They both sound kind of similar, and actually they are both nucleic acids, which are a type of biomolecule. But let's go ahead and compare and contrast them real quickly. So DNA, it stands for deoxyribose nucleic acid. The deoxyribose is a sugar, and nucleic acid is the type of biomolecule that it is. DNA is also double-stranded. That means it has two strands, and it's in a double helix shape, also known as a twisted ladder. nucleus are adenine, thymine, cytosine, and guanine. And they go together like this. If we remember the little mnemonic device, apples in the tree, that helps you remember A goes with T, and car in the garage, so C goes with G. Also, DNA is found in the nucleus. So those are all things about DNA. RNA, on the other hand, stands for ribonucleic acid. The ribo, that's actually part of ribose, which is a sugar. So just like DNA had deoxyribose, RNA has ribose. They both contain a sugar. RNA also has 4 bases. Remember in DNA, they were A and T, C and G. Well, in RNA, you have all the same bases except for one. There is no T, otherwise known as thymine. There's no T in RNA. Instead, it's a U. The U is for uracil. So you can't remember apples in the tree for RNA. That's not going to work because there's no T. So instead, try remembering apples under. So it's kind of like they're under a tree. A for adenine and U for uracil. They go together. Also, there's still C and G. Remember cars in the garage, that helps you remember cytosine goes with guanine. So the bases are pretty much the same in RNA except for the uracil instead of thymine. RNA also will start out in the nucleus, but it's going to travel out of the nucleus. It's going to help deliver the message. There are actually 3 types of RNA. But don't worry, because what they stand for, it really helps give away what they do. And let me give you an example. So the first type of RNA is messenger RNA. And the actual acronym that you'll see for it is mRNA, messenger RNA. And its job is to carry a message based off of the DNA. Second type of RNA is called the transfer RNA, or abbreviated tRNA. And its job is to transfer the message. And then we have rRNA. That stands for ribosomal RNA. And so, kind of like it sounds, it actually is a component of the ribosome. If you remember back in cells, we've talked about how ribosomes make protein. And it's a very important thing in protein synthesis. Obviously, ribosomes are going to be involved because that's what we're doing. We're making protein. Now that you know the difference between RNA and DNA, you're completely ready to explore the concept of protein synthesis. We have another clip on that fascinating process. That's it for the Amoeba Sisters, and we remind you to stay curious. talking about DNA and how awesome DNA is, but what we have not talked about is how DNA actually codes for your traits. Say, for example, we're talking about your eye color. Yes, the DNA codes for the genetic information that codes for the color of your eyes, but how exactly does that work? Well, keep in mind that your eyes are the color that they are based on a pigment that is inside the eyes. And in order to have that pigment, your DNA must code for proteins that help make that pigment, which gives them their color. So today what we're going to talk about is how your DNA leads to a protein. And this is actually called protein synthesis. Just like it sounds, whenever you say synthesis, that means to make something. So protein synthesis means to make protein. Whenever we're talking about protein synthesis, we also need to realize that this is happening in all of your cells, okay? Because all of your cells contain your DNA. And while not all of the DNA is turned on at any given time, it's based on what type of cell we're talking about, that DNA is present in the nucleus of all of your cells. What we have to consider is, how are we going to get the genetic information that's in the DNA, which is inside the nucleus, out of the nucleus so that the cell can start producing the proteins that it needs to make based on the genetic instructions? Well, in order to answer that question, we need to go into the process of protein synthesis. In protein synthesis, there are two major steps. Something called transcription, and then something called translation. There's actually a lot of trans words that we'll get into in biology, but just stay with me here. Transcription has a C in it, and translation has an L in it. And I remember that C comes before L in the alphabet, and so that helps remember that transcription comes first. I don't know, that's always kind of helped me. Now, transcription is when we're going to transcribe the DNA into a message. And if you think about that, think where DNA is. Well, DNA is always in the nucleus. So we're starting transcription in the nucleus. And what we're going to do is the DNA is going to have matching RNA bases matched up to it with the help of some enzymes to make mRNA. So in transcription, we make mRNA. We make a message out of RNA. So, at the end of transcription, we have made an mRNA molecule. Remember, that's messenger RNA. And one great thing about being an mRNA is you can get out of the nucleus. So the mRNA goes out of the nucleus where it's going to attach to a ribosome. Remember, ribosomes make protein. Also remember that the R for ribosomal RNA, that's what makes up a ribosome. So the messenger RNA is going to go to a ribosome, which is made out of rRNA. And now what we're going to do is we're going to use the message to build our protein. This step is going to be called translation because we're translating our message into protein. And we are also outside of the nucleus. We are in a ribosome, so that's where our location is now. You know, you can find some really great clips, some animations on translation that are just fantastic. Really, what I like to think of is you've got this mRNA and it's in a ribosome, and then you have these tRNAs coming in. tRNA, remember that stands for transfer RNA. Well, their job is they have an amino acid on them. And an amino acid, if you remember, is the monomer for a protein. So what that means, it's a building block for protein. So if you get a bunch of amino acids together, you build this big old protein. All of these tRNAs are looking for the matching bases on the mRNA, and when they match up, they will drop off the amino acid that they are carrying. So eventually what ends up happening is you get this big chain of amino acids that have to match up with the coding on the mRNA. The different amino acids are held together by something called a peptide bond. It's basically a protein bond, and you end up at the very end with a really long protein made of all these amino acids, which now can be used for all kinds of different body processes. And that could be something, for example, if we're going to make eye pigment. One important thing to mention is that when the tRNA is bringing in the amino acid, It reads the letters on the mRNA in threes, meaning every time you have 3 bases, that's how it reads it. So it doesn't read one letter at a time, it reads it in triplets, in groups of 3. That's called a codon. A codon is made of 3 bases. So, for example, if you had an mRNA strand, it wouldn't just read A, and then U, and then G, and then some other letter. It would read AUG together, a triplet, a codon. And so because it reads in threes, when we get into mutations, we'll talk about how if you end up taking out one letter, that could be very bad, because if you read in threes and you take out one letter, that will mess up your reading frame for the rest of that strand. So when we get to mutations, we'll really go into that. One thing I'd really like to encourage, with topics like this in biology, it's really great to have some kind of activity so that you can really walk through it. Because it's really hard to visualize this protein synthesis, but if you can do an activity where you're actually involved in it, it will help it make a lot more sense, I promise. So if you do a Google search for PBS DNA workshop, just all of that, you will pull up this great resource. The PBS DNA workshop not only has a little workshop of DNA replication, but it also has a great protein synthesis activity where it will actually let you do the protein synthesis part, both the transcription and the translation. Just highly recommend it. Anyway, that's it for Amoeba Sisters, and we remind you to stay curious.